Good afternoon, everyone. My name is Kellan Moore, and today I will be presenting on the microRheology project I worked on this summer. MicroRheology is a useful technique to non-invasively probe soft matter at the nanoscale to gather information about its structural and mechanical properties, such as viscoelasticity. It is especially useful when characterizing complex cellular microenvironments because it is able to capture the heterogeneous nature of the sample of interest. These findings have important ramifications in the design of complex biomaterials and nanoparticles in the advancement of drug delivery systems and tissue engineering. This summer, I worked with mucus samples. The research question that I was investigating was how can microRheological techniques be used to characterize complex biological fluids, such as mucus, as well as how do those results compare to one another and to bulk rheology performed with larger sample sizes. One of the techniques that I used this summer was multiple particle tracking, which is a passive way to gain information about a sample's mechanical properties. I placed 1 micrometer fluorescent beads in my sample and recorded their resultant Brownian motion. From this, I exported the data to a software system that provided information about the displacement of each particle. This was then used to solve for the diffusion constant as shown in the equation below. The only unknown after determining this constant was viscosity, which was the property of interest. After using this method to calibrate with glycerol, I moved on to characterizing my synthesized mucus samples. Here are the results that I gathered from this method. Typical mucus contains 3 to 7% protein, so I used 5% mucin, which is the main glycoprotein in mucus. The 1% albumin was used as a representative of the other proteins present in naturally occurring mucus. I also included varying concentrations of glutaraldehyde, which is a cross-linking agent. This means that it stimulates mucin proteins to bind to one another, resulting in a stiffer solution. It was expected that with increased glutaraldehyde concentration, there would be a decrease in the average displacement of the particles, as well as an increased viscosity. This trend can be observed in the plot to the right. In conclusion, increased glutaraldehyde concentration resulted in a decreased mean squared displacement and an increased viscosity, indicating the viscoelastic fluid became stiffer and behaved more like a solid. Future work can expand on these findings by testing varying amounts of mucin and glutaraldehyde concentrations to determine if the trend holds. Additionally, active microRheology can be performed and the resultant viscosity can be compared to passive techniques. Finally, this work can be expanded to other biofluids such as serum. The characterization of biofluid viscosity is important to the field of biotechnology, especially in the design of nanorobotics so that they can easily navigate through their cellular environment to provide targeted drug delivery. Thank you very much for listening to my presentation.